Hello students, let's continue the next part of the lecture. So in the t-test we have seen in the last lecture that t-test is a parametric test and the data for the t-test you should assume that the data is independent, normally distributed and have a similar amount of variance within each group being compared. So if your data is not fitting in these assumptions, then you should use non-parametric test, which is alternative for the t-test. Now, one or two sample or paired t-test is there. There are different types of the t-test. If the group come from a single population, that is measuring before and after an experiment, then you can perform a paired t-test. This is a within subject design. So if the group come from two different populations, that is two different categories or two different species or people from two separate cities, then you should perform a two sample t-test, that is independent t-test. And this is between subject, again this is between subject design. This was within subject design because you are just doing the same experiment, just the measurement is before and after. Here you are taking two different groups altogether, so it is between the subject design, right? So if there is one group being compared against the standard value, that is comparing the acidity of a liquid to a pH that is normal or a neutral pH 7, so then you can perform the one sample t-test. So these are the different t-tests you can perform as per your requirement, that what exactly you are doing. So if you are taking measurement of the experiment before and after, you must do pair t-test. If it is between the subject group, then you should do two sample t-test. And if you are just comparing against a standard value, then you should do one sample t-test. So there is again one-tailed or two-tailed t-test. We are not going to go in detail about the statistics, as it is even I said it previously also, it is expected to be taken by your department itself at department level. So if but still I'm just touching few points so that you could relate it with whatever you learn in your department RM. So one-tailed or two-tailed t-test, if you only care whether the two populations are different from one another, then you can do two-tailed t-test. So there should be two population different from one another. And if you want to know whether one population mean is greater than or less than the other, then you need to perform one-tailed t-test. So whenever you are just finding out these differences, or what is your purpose of the data, what type of data analysis you want, accordingly you have to choose this one-tailed or two-tailed. So for example, in your test of whether that petal length, which we have seen in the last slide, that is in your observation that petal length differences by the species, your observation come from two separate populations. So you perform two sample t-test, as your observations are coming from two different samples, right? Two different species, because even though the plant variety is same, species are different. And you don't care about the direction of the difference, only whether there is a difference. So you choose to use the two-tailed t-test. It depends, even in the same experiment, you can try to use the different t-tests also. Now, what is the formula of t-test? The formula of this is formula specially for two sample t-test, that is t is equal to the formula is T is the T value. X1 and X2 are the means of the two groups being compared, that is X1 minus X2. S2 the pooled standard error of the two groups, and N1 and N2 are the number of observation in each group. So S2 then S square actually 1 upon N1 plus 1 upon N2. So the larger T value shows that the difference between two group is greater than the pooled standard error, so which indicates a more significant difference between the two groups. Now the next is Z-test. Now Z-test is again statistical test to determine whether two population means are the different when the variance are known and the sample size is large. A Z-test is hypothesis test in which Z statistics follow a normal distribution. Now a Z statistics or Z score is the number which represents the result from the Z-test. So Z-tests are closely related to T-test, but T-tests are best performed when an experiment has a small sample size, and Z-test assume the standard deviation is known, while T-test assume that it is unknown. Now, when should you use Z-test? So, according to your data, you have to decide. So, if the standard deviation of the population is unknown and the sample size is greater than or equal to 30, then obviously you have to use Z-test. Then the assumption of the sample variance equaling the population variation should be made using the Z-test. So regardless of the sample size, if the population standard deviation for a variable remains unknown, then also a t-test should be used instead of Z-test. So what is Z-score? The Z-score or Z-statistic is the number which represents how many standard deviations above or below the mean population the score derived from a Z-test. So if the Z-score is 0, it indicates that the data point score is identical to the mean score. A Z-score of 1, it indicates a value that is 1 standard deviation from the mean, and Z-score may be positive or negative, or with positive value, it indicates the score is above the mean, and negative score indicates that it is below the mean. So there are examples of Z-test can be conducted as Z-test include 1 sample location test, a 2 sample location test, a pair different test, and a maximum likelihood estimate. So Z-tests are closely related to T-test, but T-tests are best performed when an experiment has a small sample size. Also, T-test assume the standard deviation is unknown, and Z-test assume that it is known. Now, ANOVA, it is called as analysis of variance, or ANOVA is a statistical method that separates observed variance data into different components to use the additional data. So again, a one-way ANOVA, which is used for 3 or more groups of data to get the information about the relationship between the dependent and independent variables. The formula for analysis of variance is ANOVA coefficient, that is F is equal to mean sum of squares between the groups MSB divided by mean square of error, that is MSE. Now, there are 2 main types of ANOVA. One-way ANOVA, that is unidirectional, and two-way ANOVA. So there are also variations of ANOVA is right now, that is MANOVA, that is multivariate ANOVA, which differs from ANOVA as a formal test for multiple dependent variable simultaneously you can assess. So one-way or two-way refers the number of independent variables in your analysis of variance test. A one-way ANOVA evaluates the impact of sole factor on a sole responsible variable. It determines whether all samples are same. The one-way ANOVA is used to determine whether there are any statistically significant differences between the means of 3 or more independent groups. Now we will see one video. And
हेलो दोस्तों, वेलकम टू इंग्लिश डेटा। आज हम बात करने जा रहे हैं टेस्ट के बारे में। देखेंगे टेस्ट क्या होता है? तो बेसिकली देखा जाए तो स्टैट्स में बहुत सारे टेस्ट हैं, बट आज हम दो टेस्ट के बारे में बात करेंगे, बेसिकली पैरामेट्रिक एंड नॉन पैरामेट्रिक टेस्ट के बारे में। तो देखते हैं पैरामेट्रिक टेस्ट क्या होता है। तो चलिए स्टार्ट करते हैं। तो बेसिकली दोस्तों, पैरामेट्रिक टेस्ट में तीन टाइप के टेस्ट होते हैं। पहला होता है हमारा टी टेस्ट, दूसरा होता है जेड टेस्ट एंड थर्ड वन इज एफ टेस्ट। तो चलिए स्टार्ट करते हैं वन बाय वन। पहले हम देखते हैं टी टेस्ट के बारे में। तो बेसिकली फ्रेंड्स, जो टी टेस्ट होता है वो एक स्मॉल सैंपल टेस्ट होता है। ठीक है, जब हमारा सैंपल साइज स्मॉल होता है तो हम टी टेस्ट का यूज करते हैं। इट वाज डेवलप्ड बाय विलियम गॉसेट इन 1908। इसको डेवलप किया था विलियम गॉसेट ने 1908 में। और इस टेस्ट को हम स्टूडेंट टी टेस्ट भी कहते हैं, क्योंकि ये एक पेन का नाम था जिसके थ्रू ही ड्राइव्ड हुआ नेम स्टूडेंट टी टेस्ट, जो कि ड्राइव किया था विलियम गॉसेट ने। इसका फॉर्मूला होता है स्टूडेंट्स, एक्स बार माइनस म्यू जीरो अपॉन एस अपॉन रूट एन। इट मीन्स जो यहाँ पे हमारा ये एक्स बार है, ये हमारा सैंपल का मीन है, और जो म्यू जीरो ये हमारा पॉपुलेशन का, टोटल पॉपुलेशन का मीन है, और ये एस है, ये हमारा सैंपल स्टैंडर्ड डेविएशन है, और ये जो हमारा रूट एन है वो है हमारा सैंपल का टोटल पॉपुलेशन। ठीक है, तो इसके थ्रू हम अपना टी कैलकुलेट कर सकते हैं। फिर उसके बाद ये अब कहाँ कहाँ यूज करते हैं या इसके एप्लीकेशंस क्या होते हैं वो देखते हैं। तो ये हमारा जो टी टेस्ट है वो हम तभी यूज करते हैं जब हमारा जो सैंपल साइज होता है वो 30 से छोटा होता है। ठीक है, उसके बाद डिग्री ऑफ फ्रीडम निकालने का जो इसका फॉर्मूला होता है वो होता है न्यू इक्वल्स टू एन माइनस वन। ठीक है, एन क्या है? हमारा पॉपुलेशन का नंबर। ठीक है, सैंपल साइज। उसके बाद एक क्वेश्चन आया हुआ था इसके ऊपर कि सिग्निफिकेंस ऑफ रिग्रेशन ऑफ कोफिशिएंट इन रिग्रेशन मॉडल में क्या यूज किया जाता है, कौन सा टेस्ट यूज करते हैं? तो टी टेस्ट यूज किया जाता है। ठीक है, उसके बाद एक और क्वेश्चन था कि टी टेस्ट कब यूज होता है। ठीक है, तो टी टेस्ट तब यूज होता है जब हमारा जो पैरामीटर होता है पॉपुलेशन का वो नॉर्मल होता है, तब हम टी टेस्ट या टी स्टैटिक्स यूज करते हैं। उसके बाद फ्रेंड्स, इसके अंदर सबसे जो इम्पोर्टेंट पॉइंट है वो है कि इसका जो पॉपुलेशन वेरिएंस होता है वो होता है अननोन। ठीक है, उसके बाद इट इज ऑल्सो कॉल्ड वेल्च टी टेस्ट। इसे वेल्च टी टेस्ट भी कहा जाता है। कोरिलेशन ऑफ कोफिशिएंट इन पॉपुलेशन इज जीरो। टी टेस्ट में कोरिलेशन कोफिशिएंट जीरो होता है, बट इसके जस्ट अपोजिट जेड टेस्ट है। जेड टेस्ट के अंदर जीरो नहीं होता। तो चलिए जेड टेस्ट देख लेते हैं। तो जो जेड टेस्ट है ये फिशर ने डेवलप किया था, और टी टेस्ट इज यूज्ड, अब यहाँ पे मैंने वही पॉइंट लिखा है कि टी टेस्ट इज यूज्ड व्हेन कोरिलेशन कोएफिशिएंट ऑफ पॉपुलेशन इज जीरो। ठीक है, बट if population coefficient correlation is not zero, then z test will be used. ठीक है, उसके बाद z test का फॉर्मूला देख लेते हैं। तो z test का जो हमारा फॉर्मूला होता है वो होता है एक्स बार माइनस म्यू जीरो अपॉन स्टैंडर्ड डेविएशन अपॉन रूट एन। ठीक है, तो यहाँ पे हमारा जो एक्स बार है वो सैंपल का मीन है, और म्यू जीरो है वो हमारा पॉपुलेशन का मीन है, और सिग्मा जो है वो स्टैंडर्ड डेविएशन है, और एन जो है हमारा नंबर ऑफ ऑब्जर्वेशंस है। तो इसके थ्रू हम जेड कैलकुलेट कर सकते हैं। उसके बाद हम इसके यूजेस देख लेते हैं। तो इसका यूजेस जो है वो, ये बेसिकली जो टी टेस्ट था वो हमारा स्मॉल सैंपल के लिए यूज होता है, बट जो हमारा जेड टेस्ट होता है वो लार्ज सैंपल टेस्ट के लिए यूज होता है। उसका जस्ट उल्टा है, जहाँ पे हमारा जो पॉपुलेशन होता है वो मोर देन 30 होता है। फिर अनदर वेरी इंपॉर्टेंट पॉइंट, वहां पे क्या था कि हमारा पॉपुलेशन वेरिएंस जो था वो अननोन था, बट यहाँ पे क्या है, यहाँ पे हमारा नोन है, हमें पता है। ठीक है, तो वही पॉइंट है कि z टेस्ट इज यूज्ड टू डिटरमाइन वेदर टू पॉपुलेशन मीन्स आर डिफरेंट व्हेन पॉपुलेशन वेरिएंस आर नोन। नाउ द थर्ड पॉइंट, z टेस्ट इज बेस्ड ऑन स्टैंडर्ड नॉर्मल डिस्ट्रीब्यूशन के ऊपर बेस्ड होता है। z टेस्ट इज आल्सो कॉल्ड लार्ज सैंपल टेस्ट, क्योंकि लार्ज सैंपल टेस्ट में यूज होता है। नाउ फ्रेंड्स, हमारा जो तीसरा टेस्ट है जो कि है एफ टेस्ट, जिसको हम वेरिएंस रेशियो टेस्ट भी कहते हैं, या आप सिंपल सिंपल कह सकते हैं कि हमारा अनोवा है। ठीक है, अनोवा में हम एफ की वैल्यू निकालते हैं, एफ क्रिटिकल या एफ टेबुलेटेड। ठीक है, तो बेसिकली एनोवा को ही हम एफ टेस्ट कहते हैं। अगर आपने एनोवा नहीं देखा है या आपने पढ़ा नहीं है तो मैं डिस्क्रिप्शन में डाल दूंगा अनोवा, आप वहां से देख सकते हैं। टू द टू इंडिपेंडेंट एस्टिमेशन ऑफ पॉपुलेशन वेरिएंस, ठीक है। तो बेसिकली जो एफ टेस्ट है या अनोवा है, वो एस्टिमेशन हम देखते हैं कि कितना डिफरेंस है और कितना नहीं पॉपुलेशन वेरिएंस के अंदर, ठीक है। और इसके अंदर जो होता है एफ टेस्ट का, वो होता है कि लार्ज सैंपल वेरिएंस का स्क्वायर, ठीक है, से कंपेयर करना वेरिएंस, तो हम एफ टेस्ट का यूज करते हैं। नाउ
So now we will see the next part, that is data presentation. So in now you have designed, you have hypothesized, you have given the objective. After that you did applied some method, you did the experiments, and now you have a data, you have analyzed the data. Now it is time to represent the data. So data representation is an effective tool for resolving information issue. So basically, whenever you want to represent the data, it will create a clear idea or clear picture about whatever research you have done, so that the front person will understand it in a the front person will understand it in a better way. Now, what is data representation? Data representation is how data is represented or presented to users. Now, for example, human languages represent physical world, that is written form, and even in the digital world, that is spoken or written form. So the data representation includes charts, graphs, table, and other tools for representing the data. So especially all these are used to represent the data in a better way. So there are 4 types, that is the graphical representation of data, pictorial representation of the data, diagrammatic representation of the data, and tabular representation of the data. So graphical representation of data, which refers to the visual display of the data. A graph is actually a diagram which gives or present the information in visual form, so such as bar graph or line graphs. And when you see towards the graph, you can immediately understand very clearly that what is the actual result, or what is the data telling you, or what is the conclusion of that particular data. So graphs show relationship and trends in data through lines, bars, and other shapes. Now another one is the second one, that is pictorial representation of the data. Pictorial representation is presented in the form of image. So this can be static or dynamic, but usually it is presented as a still picture. So the example of this type can be the image of graphs, especially bar graphs, which are used to show in data over the time or the evolution of the ecosystem. Third one is diagrammatic representation of the data, where the diagrammatically the data is represented. And the fourth one is tabular representation of the data, which is the data is arranged in a form of table. Of course, the table is always having rows and columns, and data is arranged in that particular format. And that we are going to see that how it is organized in the form of graph, in the form of diagram, in the form of tabular form. Now, what are the benefits of the data representation? Data representation is the key for communicating, analyzing, and sharing the data. How nicely you present the data, in that way it will get more easily accepted, because the audience will, or the other researchers will understand it in a better way, and they can accept it easily. So different types of data representation that we saw earlier allow us to interpret and understand the data. Earlier, in the sense, these 4 data representation which we have just seen, those will help us to interpret and understand the data in a better way. So data can be represented in various forms that highlight relationship between the components. So whenever you are seeing a data, you can easily, immediately figure it out that there is what type of relationship between the two components. Even the variable or independent variables, you could able to easily figure it out and make the data easier. So the way you are presenting, it will make the data easier to interpret, right? So anyone, the one who is sitting, if you give them in writing the whole synopsis or whatever you did, instead in their hand, and just present the same thing in the form of either a graph, either a table, either diagram, they will understand that more easily, that representation of data more easily than reading the whole things, right? So that is actually the importance of the data. So some of the benefits of the data are as follows: better comprehension of data, that is, data can interpret it visually and can help us on get an overview of the information presented. Then better decision making ability. Once you see the data clearly, then it can immediately clicks you that yes, this type is done, this, there is some problem, you can correct it, you can add this. So that's why in research, while doing PhD, after each 6 months there is a presentation, and there you need to present your data in the form of graphs or in the form of tabular or in diagrammatically pictorial form. You need to present it so that those experts which you have called to analyze them, they will analyze it critically and tell you the problems, and accordingly you can do the correction and go ahead, right? So in research, while doing research also, you can do the correction and go ahead. So better decision making ability it gives. Then predictability of the data can be presented in different forms and make it easier to interpret. So this means that if you have a data on a piece of paper, as I said, and use the same form in a visual way, it is more easily you could understand the data which you are presenting visually. So data sharing and communication become easier. So once you have represented the data in a particular format, which we have seen before in the last slide, that this makes the data sharing easier. Now I instead of sending you big files, I can just send you some pictures of my data, pictorial form or in a tabular form, and it's done. You will understand everything well. Now, as we have seen 4 types, now in graphical representation, you can present the data in different types of graph. So these graphs will help you to, because it is like when you have to draw which type of graph is very important. Everywhere you cannot. So whatever data you have, you cannot just draw one type of graph only. You cannot do that. So there are different types of graphical representation. Those are line graphs, bar graphs, histograms, line plot, frequency table, circle graph, stem and leaf plot, and box and whisker plot. So the line graph or the linear graph is used to display continuous data. So what kind of data is getting represented, it depends on what type of data you have. Accordingly you have to choose the graph. So whenever continuous data is there, that something is growing, that microorganisms are continuously growing, so you have to show that data in a, or the enzyme which is getting produced, it is increasing continuously, you have to show it in a, it is whatever is there, it is happening continuously, then you have to draw the line graph. And it is useful for predicting the future events over the time, because it will grow or it may not grow. In case of bacteria, after some time it will go to the stationary phase, so straight line will be there, and again it will drop down, so because the bacteria will get died. So at the line graph will represent it in a better way. Now the bar graph is used to display category of data. right? Whenever the data is divided into different categories, at that time bar graphs are the best one, because it compares the data using solid bars which represents the different quantities of the different categories. So it is always better whenever your data is categorized, at that time you have to use bar graphs. Histograms, the graph that uses bar to represent the frequency of numerical data. So whenever numerical data is there, which is organized into intervals, at that time you have to use the histograms. And as these all intervals are generally equals and continuous, all the bars have the same width, so automatically you will get the similar width. Now the line plot, it shows the frequency of data on given number line, right? So x is placed above a number line each time when the data occurs again. Then frequency table, the table shows that the number of pieces of data that falls within that particular given interval. Circle graph, it is also known as pie chart, which shows the relationship of the different parts as a whole. Means it is the whole picture, and the different parts like pizza pieces are there, different when you cut the pizza, different pieces are there. So like that, it is having different parts, but the whole pizza is only one. So like the whole picture will be only one. The circle is considered 100%, and accordingly you can have the different pieces, right, which indicates the different percentages. Now stem and leaf plot, the stem and leaf plot, in this particular plot the data is organized from least value to the greatest value. That's why it is called a stem and leaf plot. The digits of least place values from the leaves, and the next place value digit forms the stem. So least place values are the leaves, and next, that is, you can say it goes to higher, that forms the stems. Now box and whisker plot. Now this plot diagram summarizes the data by dividing into 4 parts. Box and whisker shows the range and the middle of the data. So these are all the pictures of those graphs. So this is the bar graph. This is histogram, which looks like a bar graph, but there is a difference. Then frequency table, right? Then this is the circle graph. This is also called as pie graph. Stem and leaf plot. This is the leaf, and this is the stem. Then this is the line graph, line plot, and this is box and whisker plot. Now we need to stop it here. We will see the next graphical representation in the next lecture. Thank you.